Yeah, this is kind of experimental. I guess I'll just jump right into it and just go with it.
Yeah, I don't even know. So I guess to start, we may have to re-tape all of this because
Sure. Yeah,
I'm not sure that this mic is pretty good, but I don't know if it's that good. And I ordered a much longer cable that, like next week, I can kind of put the mic right in the middle of the table.
Yeah. Yeah. Thanks, Timmy. I'm happy to get the practice, and it's cool to just develop the skill in doing this. Okay, so welcome to the Cleveland Cyber Higher Education group. Today we're talking about bioinformatics done on an exciting integrated platform called BioBike. What is bioinformatics? Bioinformatics is the study of information in biology. There's different branches of it, different areas of concern. The area of concern that we're going to look at today is molecular bioinformatics, genetics. This is such a happening field. It's exciting. What we do in molecular bioinformatics is that we study genes and proteins. Proteins are coded by sequences in genes of amino acids. So it's a very good topic, a very good area for the application of artificial intelligence. Biologists have studied genes, they've studied proteins, they know the properties of many proteins, and one of the main whole points of molecular bioinformatics is to predict what the behavior of a protein is going to be from a gene. So if you have a new bacteria, or you're studying some part of even the human genome that we haven't fully figured out yet, we can look at the sequence of those amino acids, and we can compare them to what we already know about other proteins, and use that information to try to predict what this new genetic sequence is going to do. Genes, genomes are big. There's thousands and millions of amino acid sequences. So there's huge databases that are needed to search through to match up one gene sequence that you're studying with a potential gene sequence in the database. And there's various tools to do this. The most popular tool is this tool called BLAST. It's an acronym that is partly run by the government, and it's available for free on the internet. Typically, the way biologists and bioinformatics people work, they'll get the gene sequence from the research. They'll start looking up information about it using BLAST, finding similar proteins, and this can be a complicated process. Some proteins will be somewhat similar. Some proteins will interact with other proteins. So they end up with this kind of tree of information that is pretty challenging to figure out. And it often calls for programming, because they'll want to compare one sequence to a number of other sequences. They'll want to find out how many times and where a certain sequence occurs in what other organisms or even in the same genome. There's two common ways they end up doing this. We'll make it three common ways. They'll have a bacteria, or they'll have some organism, maybe a plant or something. They'll pay a company to sequence the genes in that organism. The company will put the information in a multi-terabyte hard disk. They'll make a database out of it, and they'll also set up a certain amount of kind of automatic analysis systems with the database. They hire biologists to kind of go through the material they produce to a certain extent to do a kind of basic analysis on it. And that basic analysis will get about 80% of the analysis job done. But it's basically almost never enough to get fully done what you really want to do when you're trying to understand what's happening with the genome in an organism. So even though there's companies that will do a lot of this analysis for you, you end up having to do a lot yourself. And the amount of data you have to work with is way too much to work with in any way other than programming. Biologists in the past have often used things like Excel, even writing things down, and that quickly becomes unmanageable. They'll bring in a programmer, or they'll bring in someone who studied bioinformatics, which is kind of like a bioinformatics programmer. They'll apply tools like Perl, probably the most popular, to carry out their data processing tasks. But there's an issue with that, and that's that then you get a situation then where the biologist has to tell the programmer what he wants, and the programmer is probably not going to understand biology at the same level as the molecular biologist, so there's this kind of impedance mismatch. So there's a need to have the biologist have a greater ability to use automated tools easily, and there's also a need for the programmer to be able to have an easier ability to communicate and work with the biologist. And this problem is a pretty big deal, because if we can solve this problem, we can start making massive leaps towards curing cancers, towards curing neurodegenerative diseases. It's one of the most important things in biology and medicine. It's like it's right up there with protein folding in being a bottleneck in steps forward. At least that's the way I see it. And one part of this is that I kind of get the impression that many biological researchers aren't aware of the power that a truly good programmer can bring to their data processing. They underestimate what we can do. And part of that is because in the past, when they've asked us to do things, we haven't fully understood what they're asking us, and thus we're not fully applying what we can do to what they want. And another thing that many biologists may not be fully aware of is what we can do with artificial intelligence and machine learning. Using the techniques from those two areas, we can do tremendous automatic data analysis. We can make all kinds of new discoveries on the computer using those two things, themselves. And those areas are kind of obscure, and not everyone really understands what you can readily do with them. So, long story short, there's this tremendous potential in information processing in biology that is, we're only starting to scratch the surface, and there's a huge need to move forward with it. So how can we best address this issue of a need for better communication between the biologist and the programmer? BioBike. Thanks. BioBike combines multiple common tools that the biologists use, like BLAST, other programs that match sequences in phylogenetic trees to try to find family relations in organisms, and some other tools. Setting this stuff up is a real pain otherwise, because you have to set up different data formats, different databases, and you have to work with obscure stuff in usually Perl. So just putting all those together into one platform that alone is a huge deal. But it's a lot more than that. It also allows you to program the system using diagrams. For example, if I go to sequence of, and I'll type in an organism, and then run my diagram by double-clicking on the title, it's going to block my pop-up. So I close this, and then I'll just
go
read it. I'll allow my pop-ups, and I'll run it again. Okay, and now here we have the sequence of this organism. The part in red is a gene, and you can see it's got the description of the gene here. Not every gene or protein is described. That's one issue in dealing with this stuff. And we can scroll through the whole thing and see all kinds of genes. So that alone is a powerful tool. And there are commercial systems that do things like this, but BioBike is open source. And if we can go down to this window, click on View Code, and it's actually going to show us the Lisp code for that command. That brings us to our other aspect of BioBike. It's a Lisp platform. Lisp is a currently, unfortunately, largely forgotten about language, but it's also, the way I see it, the most concise language, meaning it's the most powerful. With Lisp, you can craft your code to match the problem much better than in other languages. This is because Lisp is more or less based on, or at least closely related to, lambda calculus, which is a mathematical system for describing functions. Early in the century, Alfred North Whitehead and Bertrand Russell worked on their Mathematica series of books, where they attempted to base all of mathematics on functions. And that started a series of work in mathematics that other people contributed to. And a guy named Alonzo Church came up with a way to represent basically any type of mathematical object you want with functions. Now that early attempt basically failed. They were unable to get everything in mathematics to be expressed in functions. But they basically got enough of it in there that, for all intents and purposes, for any normal math object, you'll be able to do it with lambda calculus. So, because of that, you can use this to be changed so that it pretty much exactly matches whatever problem you want to deal with. And you can also make your own commands with it. So, unfortunately, my PDF showing an example with Perl is on my other computer. But long story short...
Oh, it's on my desktop.
Oh, it's on your desktop? Great. Okay, let's see. How do I get to a desktop on a Mac?
It's double click the top bar there and minimize it. Yeah, there you go. Just minimize it. It's up on the top left there.
Right here.
Just minimize all those. Minimize that guy.
But I'm not used to this mouse pad.
Yeah, so he said,
like that part. Okay. Yeah, substitute, I know everything we can have. Just kidding. So, all right. Okay, so all right, so first we have the English language version. Find 100 base pairs of sequence upstream from a set of orthologs for all genes in an organism and align them. So that's a typical small type of bioinformatics problem. They're basically trying to find matches in a sequence under certain, under a certain set of limitations. So they'll put in programs like this to try to understand what's going on with an organism and how it works, how its biochemistry works. So we have the first version in Perl, and I've got a fair amount of experience with Perl, but you know what? To me, this looks totally confusing, more or less. I mean, I know what the do loop is, and I can see he's got the TR in there, but I'd probably have to look stuff up like TR-D. That is not on the top of my head. And then you can see he is dealing with a .fasta file, which is a type of file format used in bioinformatics. So he's trying to get Perl's set of tools to kind of form itself around the problem, and then he's dealing with an outside data format. Now if we look at the Lisp version, you can see for each gene in genes of whatever organism you're going to put in there, as the orthologs are the orthologs of gene, as the upstream sequences are the sequences upstream of orthologs with a length of 100, collect the alignment of the upstream sequences. And just looking at that, you can see that it's a much closer match to the English language and basically easier to understand. The Lisp version of this is using macros, which make a kind of special form in Lisp. The special form in Lisp causes the parts of a command to be done in a different sequence than they're normally done. The way Lisp normally works, you would have plus 2, 2, and that's going to add 2 plus 2. But for example, this for each command, it's a special form, meaning it's going to work a little differently. And I'm just pointing that out because the real power of Lisp is that it's in the form of lambda calculus, which means it's in a tree format, meaning it's reprogrammable. The point of that is that you can make special languages with it, like for example, the for each command. So you'll see stuff in Lisp programs that some commands like for each will not follow the typical prefix notation of Lisp, but they're there to help you out for a specific problem. So the power is that you get to program something that just makes a lot more sense and is a lot more applicable to your problem than Perl. A lot of people are kind of scared of Perl. Like I was talking to someone recently who I mentioned something about Perl, and they said, "Oh, I don't know anything about Perl," but previously they had explained all the stuff they knew about bioinformatics. And the way I see it, the bioinformatics knowledge is a lot more powerful and deep than Perl knowledge. Sorry, Perl guys.
There's no Perl guys.
But long story short, that knowledge in Perl, it's basically like esoteric knowledge, like technical stuff that is not really it's not really mathematically integrated. To put it that way, there's not a common base to it. It's like a toolbox that's developed over the years that people have learned apply to different problems, But if you're not aware of that toolbox and what you use it for, and then what are you going to do with it? And the only way you can really get aware of that is by spending all kinds of time on it, chopping up, text files on Linux. But Lisp does have a consistency to it. It's got a mathematical consistency. Once you learn the rules of it, you can craft it to apply to your problem. So Lisp is a powerful, forgotten language. Perl kind of took over the bioinformatics space in the 90s because Lisp was kind of outmarketed by Perl at that time. And so because of that, most of the bioinformatics people today, they do things either in Python or in Perl. So Lisp has kind of been forgotten. But despite the fact that it's been forgotten, there is no other integrated platform like BioBike. In other words, the most powerful bioinformatics platform in the world today is BioBike, a Lisp platform. So that's the basics of BioBike, what it is and why you want to use it, and why it's better than other bioinformatics packages. And I'll just add another quick factor. Lisp at one time was the most popular artificial intelligence language. That's kind of debatable now, but there are a lot of artificial intelligence packages available in Lisp. The old code for the main artificial intelligence book is written in Lisp. And the way I see it, that makes this platform very amenable to have artificial intelligence and even machine learning code plugged into it. Lisp is set up in such a way that you can easily plug stuff into it. It runs in memory. It's a running image. You can modify this image while it's running. You can add stuff to it. And so because of that, I think it's the ideal tool to take the next step into artificial intelligence for bioinformatics. And that's really the step that's needed, because there's just too much data to deal with. And the inventors of BioBike have started taking that step. They've worked on plugging in an automatic deductive logic system called SNARD into BioBike. And they are involved in running a company called CollabRx that works on cancer and other problems, and is promoting this new kind of open science, open research approach to cancer and other problems. So it's a pretty exciting field, and I would say, hey, check out BioBike, because it's an integrated platform, and it's going in the right type of direction that the other tools are not. So thanks for your time, and check back next week. What do you guys?
Well, that was good.
Thanks.
I think we may have to get. I was imagining like a wireless mic.
Yeah.
Yeah. It's something that you can kind of just pin to your lapel.
Should I cut this? Sure.